to the computer. So hi everyone, thanks for joining today. So you know, it's really nice. We have BioCurate talking today. So just as a bit of background, BioCurate was established in 2016 as a joint initiative between Monash and the University of Melbourne, with seed funding from the Victorian State Government. And the reason for that was to really help to promote the commercialisation of translational research at the two institutions. Today I'm introducing Damien Bates, who's the acting CEO and also the Chief Scientific Officer of BioCurate, and also Andy Allen, who's the Associate Director of Project Management. And today they'll jointly discuss BioCurate's approach to the evaluation and funding of early stage translational research. So look, I won't take up any more time and I'll hand it over to, is it Andy speaking first today?
I think Damien will speak for 5 to 10
Damien?
and myself.
Yeah. Fantastic. And look, I should just add that both Damien and Alan have both industry and academic experience. So, for example, Damien's been at Baxter and Allergan, just to name a few, and certainly Andy was at Pfizer. So both people have had a foot in both camps and know how things work in both of the environments. So without a further ado, I'll let you take over. Okay.
Right, thanks, Andrew. And just thank you very much for the opportunity for Andy and I to present to you all today, and for the introduction of BioCurate. And also, Mary, thanks for helping us set this all up. So as Andrew mentioned, BioCurate was formed back in 2016. It's a joint venture between Monash and University of Melbourne. And the whole idea is to try and improve Australia, and in particular Victoria's track record in the commercialisation of research. It goes without saying that Australia is extremely well regarded in terms of its research capability, and is always ranked very, very highly. But on the commercialisation side, we don't rank as highly, and there's an opportunity for improvement. So the vision was to try and set BioCurate up and provide sufficient funding. Andrew mentioned the funding totaled $60 million, which is a lot of money. And the aim was to put together a team that had industry experience, that could identify projects, science that was being conducted at both universities that had the potential for commercialization. And I'll go into that a little bit more depth in a couple of slides, but I think that's the key aspect to make sure everyone understands. There's obviously fantastic research going on. Not all of it has commercial potential, and there could be a whole variety of reasons for that. It could be that there's no real unmet need out there, there's intense competition, maybe the clinical development path is just too long and too expensive, maybe the regulatory path is not clear, or the IP landscape is messy. So lots of different reasons. So not every great scientific project has the potential for commercialization. And what we try and do is leverage our experience in the industry to try and identify those that do have the potential, and then work with you to potentially in-license them and then develop them. And the goal being to try and get a product to market that will ultimately improve the quality of life. That's what motivates us all. Andrew mentioned some of our industry experience. And maybe if we could flick to slide 3, Andy. Most of us have developed or done our scientific training in Australia. For instance, I did my PhD at the University of Melbourne in the Murdoch. Way back, I did my clinical training in Melbourne in surgery before heading off to the States, and then spent the bulk of my career, now getting close to 20 years, in industry. And I've worked in large companies like Baxter and Allergan that Andrew mentioned, but also some small startup companies as well in the stem cell space and in combination products. But overall, this is a snapshot of the team. We've all had a lot of industry experience, and we try and bring that experience to bear on the evaluation of the projects that we get in from you, and then use that experience and that knowledge to help develop those projects in a way that the regulatory authorities, whether that be the TGA or the FDA, requires in order to get what we call an IND, which allows us to start initiating clinical trials. So you can see the team there collectively has quite a lot of combined biotech and pharma industry experience. We've been involved in getting a lot of scientific projects to the IND stage and then on to NDA, which is the drug approval stage, or in the case of biologics, BLA approvals, mostly in the US, but also in Europe and in Japan. In addition to addressing these unmet needs, the market pays a lot for this. And you can see sort of the deal values involved because bringing something that's novel to the market and saving or improving people's lives is something that the payers are willing to pay for. We go to the next slide, please, Andy. This is a snapshot of, so BioCurate's been in operation since 2016. We have reviewed, I think now, it says 130 there, I think it's more like 150 projects. The percentage of projects that historically we've invested in is around 10%. So that out of 15 of those 150 we've invested in. And then all those projects have complicated development plans with clear go, no-go milestones. And not surprisingly, not every project meets its go, no-go milestone. We do an experiment. We say in order to proceed, you need to hit such and such an outcome. And we can't always predict science clearly. And so some projects can't move forward depending on what the results reveal. We have 3 projects in our portfolio currently, 2 in oncology. One's an antibody project, and that's actually reached a very exciting stage. Enough progress has been made on that particular project to justify formation of a company, which we've done, called BioTX. And there's a brief description about that asset there on the right-hand side. Essentially, it's an antibody, it's an agonist antibody that targets IL-21. And the whole point of this is to try and get around some of the dose-limiting toxicities that were observed with IL-21 in earlier clinical studies, and to ultimately try and make cold tumors hot, make them responsive to checkpoint inhibitors, and to also improve the efficacy with checkpoint inhibitors that are currently on the market. So that's at a very exciting stage. We have another cancer project that's earlier, it's a small molecule. And we have a small molecule project in the neuroscience space targeting epilepsy. And that's also pretty exciting because we recently discovered an association with that target and a form of pediatric epilepsy, which is an orphan indication, which is exciting because that provides an expeditious regulatory path. BioCurate has collaborated with a number of large pharmaceutical organizations like Novartis, Servier, Gilead, AbbVie, Takeda, and they review our projects on a periodic basis to provide feedback and input. And that's very valuable because that can direct some of the work that we do to make sure that we're producing the kind of data that they're interested in, all on the path to getting to an IND. BioCurate's also been involved with the federally funded BTD program, working with MTP Connect and UniQuest and NDPP. So yeah, that's where we're at currently. One of the big shifts that we've made recently, and that's the main reason for talking with you all today, is historically we've reviewed potential projects on as they come to us in their current form, and so we evaluate them through very specific criteria, as I said before: unmet needs, commercial potential. Are they differentiated from products currently on the market or in development? What's the IP landscape, etc. And if they didn't sort of tick all the boxes, we'd say, look, great science, but we're not able to invest at this point in time. What's changed now for us is that we want to try and make the pipeline more robust and give you all more opportunities for investment. And so we want to work with you. We've created a separate fund that we call the Proof of Concept Fund, which Andy's going to explain in more depth, to say, look, you know, you've ticked most of the boxes, or at least most of the big critical boxes. There is potential here. There is an unmet need being addressed. There is the IP path is clear, but maybe the target is not sufficiently validated, or maybe some of your assays aren't sufficiently validated. And can we provide you with funding to address those gaps so you do become an investable proposition? So this is the big change. Rather than just saying yes, no to what's being presented, we say, okay, how can we convert something that maybe is not investable in a big way immediately, but to provide some kind of bridge funding? And so I think that's a really important change, and I'm pretty excited about that development. And to that end, we already have our first Monash proof of concept project signed off on, and that's in the oncology space. Next slide, please. So this slide just summarizes what's in and what's out of scope from the BioCurate perspective. So we call ourselves a therapeutics-based development organization, and therapeutics encompasses drugs, so typically small molecules, but also antibodies as well. We look at cell therapies. We also look at combination products. So there are sometimes drug, biologic device combinations, and so that's definitely within scope, but a purely therapeutic device, and that's regulated by a different division of the FDA and the TGA, is currently out of scope, right? So it's mainly drugs, biologics, and combination products, and we're not currently investing in diagnostics, for example, veterinary medicines or vaccines. Next slide. BioCurate is a virtual company. We don't have any in-house labs, so we often collaborate with researchers like yourselves to conduct some of the early stage work. And if that doesn't happen, we've got a variety of vendors, and Andy's put together a snapshot of that around the world, whether it's synthesizing chemical compounds in China, or doing animal studies in the UK or in Germany. We collaborate with a lot of different vendors, and so in that sense we're virtual, and we integrate all these vendors into the development plans that are put together for each project. So hopefully that provides you with a bit of a background about why BioCurate's been created, a bit of the history, where we've come from, and some of the new opportunities going forward, mainly the proof of concept funding. And I will hand over to Andy to explain that to you in more detail.
Thanks, Damien. So as Damien mentioned earlier, we used to have an ad hoc application process to BioCurate, and we've refined that over the last year or so to come up with a new method of assessing projects that relies on an application process with two rounds per year. So the first round generally happens in March, and of relevance to this meeting, we've got our next round of applications occurring in September. So we call these the early assessment, a BOA, or a BioCurate opportunity assessment. And it's a fairly simple application form, which I'll show you in a later slide. And it basically gives us an understanding of the unmet need, the scientific approach, any data that you've generated. It's not an onerous application form to fill in, but any applications must come via the Monash Innovation Office. These will get reviewed, and I'm the primary reviewer, but I do bring in key experts from within BioCurate should there be an area that isn't my area of expertise. And of course, unfortunately, it's, for some of these reviews, we might decide that there's a no-go at this particular time. We tend not to say that nothing is resurrectable from a project point of view, so re-applications can occur when some of the key issues have been addressed. But for those projects which we like the look of, we will ask the team to submit another more detailed application, which we call an EOI, or an expression of interest. And again, that will go through a review process within BioCurate. And again, it could result in a no-go at this time, but if successful, and we'd like this project to be evaluated further from an investment perspective to go into our portfolio, then we would ask the team that's assigned to the project, which would include the investigator, to put together an experimental design and, with BioCurate's help, a project investment plan. We would then take that to BioCurate's investment committee for review, and hopefully in parallel we would progress with an exclusive evaluation agreement, an exclusive license agreement, and if all the ducks are aligned, then that would enter BioCurate's portfolio. And in the portfolio, we would obviously execute the experimental plan, which may involve further target validation, hit ID, lead optimization, all the way, as Damien mentioned, all the way up to a clinical candidate with IND enabling studies. And along the way, there would be milestones, go, no-go decisions that we would take to our investment committee to, again, provide more funds to support the development activities. So this was how it looked at the beginning of the year. And we then decided that we need to address some of the failures in at the BOA or the EOI stage. And by failures, I mean gaps in the scientific data that was presented. So we instigated our proof of concept fund. And I'll go into this in more detail later, but we've got budget for $1 million over the next 2 years to invest in, give or take, around 10 projects which haven't progressed from BOA or the EOI stage, to address those scientific gaps. So again, here we would put together an experimental plan. This would be led by the researcher, and this would be specifically to address the scientific gaps that were identified at the BOA or EOI stage, with an understanding that the other criteria for success, which I will also go into later, have been met, such as the unmet need, potential for IP, and etc. So once that experimental plan has been finalised and hopefully signed off, we will generate a report and review that report after the experiments have been completed. And of course, that would potentially lead to a no-go after the review of that data, or even potentially additional POC funding. So we don't mind if we haven't addressed all of the scientific gaps at that stage. So we can go back and generate another experimental plan to, again, address those scientific gaps. But of course, if it meets all the criteria for success and the data looks very robust, then of course, oops, wrong way. We will then go to a more detailed experimental plan, and it would follow the same route, with us going through the investment committee, et cetera, and then leading a project into our portfolio. So I mentioned earlier the criteria for success, and Damien mentioned these briefly, but of course the project must address an unmet need. We really do need a defined patient population that's not being addressed by current standard of care, or not being well addressed. And of course, there must be a clear potential for commercialization and licensing. So as Damien mentioned, we will take projects up to the IND stage, but in order to take it beyond that, it will require additional funding, whether it's through VC or working with some of those partners that Damien mentioned earlier. And of course, there must be to them, obviously, a clear commercial and potential for developing that asset. So that must be well iterated in the application. The asset must be clearly differentiated, hopefully on efficacy from other approaches either on the market, or of course those in early, medium, or even late stage clinical development, and even from those that have failed in development. So here it's stressed that the application should involve or should include information that you might glean from, say, Clarivate Analytics on competitive intelligence. Obviously, there must be a verified link at the target to a human disease. And with respect to IP, so existing IP is not necessary. So if you don't have a small molecule, that's fine. What we really need is the potential for novel IP. And hopefully we'll generate that during the development plan. In relation to freedom to operate, obviously we would need a clear path to be able to execute the asset in the development plan. And what we ask here is that you do some due diligence around the asset that you've been you're proposing, such that there's no known FTO issues. As Damien mentioned, the asset should have a very well-defined regulatory and clinical path to allow us to take this asset to IND, and then others to take it to phase 1, 2, and 3 clinical trials. And of course, the asset must be supported by robust and reproducible scientific data. Again, this is where the proof of concept fund comes in. So if there are gaps in that, that can be potentially addressed through that mechanism. So this is what the application form looks like. It's pretty simple, just 10 questions with limited words for a number of them. The first 4 are pretty easy. Commercial experience is not necessary. So looking at question 4, it's nice to put down whether you've had commercial experience, so you understand what it might do to develop an asset through to the market. But it doesn't preclude you from applying, and it certainly won't preclude you from getting to the next stage if you don't have any commercial experience. And again, I won't reiterate these because I've mentioned it a few times before. Unmet need is one of the questions. We do fall down, so previous applications have fallen down at this stage. The market, the unmet need is not overly clear to us. The easier you make it for us to understand the precise market segment, the easier it is for us to review and tick that box as one of the criteria for success. If you don't know patient population numbers, that's okay. However, we do need to know that there's a clear set of patients that are not being met by current standard of care. The next question, again, it defines the experimental approach, what targets you're looking at, and maybe what modality, so small molecule or antibody or any other means. Essentially, it's question 6 relates to what your target product profile would look like. Question 7, as I mentioned, or Damien mentioned before, it's really important to show how your asset is differentiated from those either currently on the market or those being developed. And again, some applications do fall through here because it's not clear to us how it is different from approaches that have gone in the past that maybe have failed. Question 8, target validation. That might be through genetic means, either in human or in animal models. You might have used tool compounds to further validate your target, and that's where you would put that sort of information, as well as in question 9, which summarizes your key research outcomes. And again, question 10, you might leave that blank. You may not have any IP, and there's no freedom to operate issues, and a statement to that effect would be acceptable. Again, applications should be submitted via Monash Innovation, so it's probably best to contact Andrew here. And again, just summarising, what the advantages of the proof of concept fund. Obviously, it's to address the scientific gaps that might arise through the BOA or EOI application, and it's really to improve the commercial investment potential of the research that you've got. So any additional IP that's generated during that program will be owned by the university. We don't claim any rights to that. And so if BioCurate does reject the project after the proof of concept fund, you can obviously use that data to search other commercial partners and maybe have more success. Again, this gives another mechanism for you working with BioCurate. The application process isn't you submit and you get a yes or a no. We do try to provide feedback on the application to strengthen it for future applications, whether it be to us or to other potential partners. And of course, the proof of concept fund will add to that. So, you know, working with us, not only for providing funds for the laboratory to support the experiment, but also getting some sort of mentorship on the development process. And of course, you know, from us, we would like to see more applications. In the 2 BOA rounds that we've had since its instigation, we've had roughly 40 applications. Again, we would like to see more. So again, the proof of concept fund will be via invite only, so you can't apply for proof of concept funding. We will invite you after the BOA or EOI reviews to potentially submit a project plan for execution. Again, most projects must have met the majority of the criteria for success to be eligible for proof of concept funding. We would like there to be a 30-day limit to submitting your experimental plan on receipt of a desire for you to submit a POC application to us. The budget is roughly 100K, but there is a bit of wiggle room there, and we're more than happy to accept leverage funds such as the MRIF fund to support the development of that asset during the proof of concept fund. As I mentioned before, we can go back and have more proof of concept funding if we haven't addressed all of the gaps. But as has been mentioned before, progress beyond POC is not guaranteed. And during the POC fund, all of the IP, whether background or new IP, remains with the university. So we don't have any rights to that. So it's a risk for us, but it's a risk we're very happy to take to increase the quality of the asset. So I think this is my final slide. So this goes over the timing for the proof of concept, the BOA, EOI and proof of concept funding. So we're about here at the moment, undergoing this information session. We require applications by the beginning of September. So here, the 1st of September is the closing of the application date. I will review with others the applications over the month of September, and then a decision will be announced at either the end of September or beginning of October on which ones we would like to proceed to submitting an expression of interest application. At this time also, we will announce those projects that we would like to receive a proof of concept experimental plan. And again, we would like that to be returned to us within a 30-day timeframe. For those that are successful in getting an EOI request, again, BioCurate undertake to review those in 30 days. It's not mandated that you present to BioCurate, but it would offer an advantage to address any questions that we might have about the project. And again, announcements will be made at the end of November, beginning of December, for progression to the investment committee. And again, at any one of these stages where projects do not progress, either EOI, there will be a chance for proof of concept funding. So 2 cycles per year. There is the potential for projects that fall outside of this cycle, if they're exceptional projects, for us to review them out of cycle. But we expect the majority of projects to be reviewed through this 2 cycles per year. So I think that's the end of my part of the presentation. So I think Damien and I will be very happy to answer any questions that you might have, and hopefully we'll see some applications from those present and others watching this web recording by the 1st of September. Thank you.
Yeah. Oh, look, Andy and Damien, thanks for that really clear presentation. I think it was really clear, and there's a few important points. I think, we'll open up for questions. If people had general questions, they're welcome to put it into chat, but we can also take individual questions. I did actually want to just ask 1 or 2. I thought just some of the points that you made, one of them that was particularly important was that in the POC project that you described, that BioCurate does not claim any intellectual property rights to what's developed. And that was a very interesting point, because often that's not the case.
Yeah, I mean, it does give you clear potential to go and take the asset should BioCurate not be interested to other parties without any encumbrances from BioCurate. So that's an important consideration, I think.
I thought another thing that would be good to clarify too, Andy, was with the development of the experimental plan. Now, given that you're looking at some gaps and so on, is BioCurate prepared to make any input into that plan? Or is that just simply back in the...
Absolutely. Yes, we don't, during that month, we're more than prepared to meet with the researchers and to help develop that plan. We're not going to review that plan without having seen it prior to the 30-day deadline.